Well, hello there, my 112 class. This is going to be your chapter 23, Evolution of Populations. So let's go ahead and share that. You have these PowerPoints in your D2L content tab. I'm just going to go ahead and share my screen in PowerPoints with you. Actually, let's go ahead and get this set here. So chapter 23, Evolution of Populations. If you recall in chapter 22, I mentioned that individuals don't evolve. Individuals can adapt within the limitations of their own genetics, but it is actually the populations that evolve from one generation of population to the next generation of population. So oh, this is exactly what I was saying. It's a common misconception that individuals evolve. So natural selection is going to act upon individuals, but only the population will have those after effects of evolving, meaning that nature is going to select specific individuals to be the most fit for their environment at that time. And thereby, because of that selection, that is going to determine what the next generation, what their traits will be. Because if you were selected against, meaning you didn't make it, that means you probably didn't have enough kids to go to the next population, the next generation. And so, for example, a population of medium ground finches on Daphne Minor Island. So during a drought, large beaked birds, they were more likely to crack large seeds and survive. But let's go to the next. So when you look here, they were large seed eaters. And so the question is, are the finches, are they evolving? And so to look at this graph here, it shows you the average beak depth. So we're talking in millimeters up the side. And then it shows you in 1976, and also similar to the prior 3 years, so basically 1973 through 1976, on average, the beak depth in millimeters was just under 9 millimeters. And so after a drought in 1978, it is thought that the average beak, not thought, data shows that the average beak depth was greater than 9.5. So it's working its way up to 10. So the average depth of the beak was more. And so the finch population evolved, meaning the generations that followed each of those different generations, the beak depth started getting greater and greater. And so what this shows is because of the environment at the time, those individuals that had larger beaks actually tend to do well. Because think about it this way, during the time, there were lots of different sizes of seeds, small seeds, medium seeds, and large seeds. So if you had a smaller beak depth, you ate the small seeds. If you have a larger beak depth, you ate larger seeds, but that didn't stop you from eating medium seeds, and it didn't stop you from eating small seeds. Now, don't get me wrong, probably a little bit harder to round up the small seeds, but you can still round them up. Versus on the flip side, a finch with a smaller beak depth, they're limited to eating the smaller seeds. So after a drought, what that did is that changed the environment. And so those bushes, those trees that only made the smaller seeds, the medium seeds, they must have suffered during that time. Thereby, those individual finches that had larger beaks, they were able to eat more food. And since they were able to eat more food because they could eat the large and they can eat what's left of the medium and the small, then they were able to essentially get more nutrition. Getting more nutrition, that is going to add to your growth during your growth periods of coming from adolescent into adults. And guess what? Those that had the genetics, because the matter of growth is not only a genetic factor, but it is also a state of your environment too. You're limited within your genetics, but if you have good nutrition, you are going to grow to the maximum potential of what your genetics will allow. And if you eat well, that means you are going to have enough nutrition to have really good reproduction. You will find in populations in general, if the organisms of a population are not eating well, let's try that again, got a phone call. If the organisms of a population are not eating well, what will happen is reproduction is going to be the first to go. They're not going to put the energy that they actually have, because that's what eating is about, acquiring energy. They're not going to put that energy, that they're acquiring from their food, into reproduction. And this is not a conscious decision. This is a natural thing that takes place. Reproduction is always going to fall by the wayside in starving populations. Okay, so with that, in the smallest unit of evolution, we're going to call this microevolution. Microevolution is the change in allele frequencies in a population over generations. And so if you recall, when we talk about alleles, you learned about alleles when you did your Bio 111 class, specifically when we went into your genetics piece. That would have been beginning in chapter 14. It started talking about Mendelian genetics, but it didn't really refer too much to alleles, because Mendel at the time didn't know about alleles. And then chapters following that, they started taking Mendel's concepts and applying them to the alleles. So as a quick little reminder, let's just go ahead and pull my pen out. So if you recall, I'm going to go ahead and draw a nucleus. Ooh, could have been a better nucleus, right? If you get one chromosome, we're going to call this chromosome number 1 from one parent, and then you get another chromosome. Let's go ahead and get another pen color. You get a second chromosome from the other parent. And I have no clue what happened, but we'll redraw it. So here's the nucleus of an individual. Here's a chromosome. We're going to call it chromosome number 1. You got it from one parent. Then you got a homologous chromosome number 1 from the other parent. So one of these came from the mom, one of them came from the dad. So these are homologous chromosomes in the fact that they have the same types of genes on them. And when I say types of genes, if you think about it, we can talk about toe length. Okay, so in this section on your chromosome, we're going to say this is the section that codes for the length of your toes, whether you have a long second toe or whether you have a short second toe. Okay, this one, same exact spot, because that is the locus on the gene, is going to code for the length of the toe, whether it is long or whether it is short. Okay, so that's what I mean by homologous chromosomes. They have the same type of genes on them. So from dad, maybe you got a long toe, and then from mom, maybe you got a long toe, the genes that code for long toes. So with that, these little sections where the genes are for the long toes, they call these versions alleles. Sometimes alleles are the same, meaning long-toed allele, but sometimes the alleles are different. So let's just go ahead and get that little eraser. Let's make a different allele on there. I'll go ahead and make it in, I guess, cyan. Okay, so, oh, well, I guess that's an okay color once I color it in. There you go. So it's in the same locus, same location on the chromosome, but it is a different version. So this one on the left, that is an allele that codes for a long second toe. And this one here, that is an allele that codes for a short second toe. So with that, microevolution is going to have a change in the allele frequencies in a population over the course of generations. So there are three main mechanisms that cause allele frequency change. That's going to be natural selection. That's something that we talked about in the last chapter. We're going to learn about two other ones in this chapter, and that is genetic drift and gene flow. So only natural selection consistently causes adaptive evolution, meaning organisms can adapt to their environment, but they're limited within what those genes will allow them to adapt. So that's something that we've already covered. So with that, the genetic variation is what is going to make evolution possible. So variation in those heritable traits, right? They're inherited from from a parent to an offspring, when that offspring becomes a parent to its next offspring. So the variation in those heritable traits, it is that prerequisite for evolution by natural selection. What this means is that without different types of traits, evolution would not take place. What we're going to find out that is a population is going to be its healthiest when there is a lot of variation in the traits within a population. If you are finding a population is it's having less genetic variation, that can actually be an issue for a population. It can cause a population to not succeed. And so Gregor Mendel, what he did, he's worked on pea plants, and that provided evidence of what he called discrete heritable units. Well, he called them discrete heritable units because he knew they were inherited from a parent to an offspring and then to their offspring and so on. And they're discrete because he didn't really know what they were. He had no clue. We now know what they are, and we know that they are called genes. So in this genetic variation, genetic variation is going to be different, hopefully, among individuals, obviously. Among individuals, it's going to be caused by different genes or DNA segments. This is a good thing, okay? So for example, I, as a human being, I make proteins. My dog makes proteins. You might find that between the species, we have similar, like the same proteins, but you may also find between the species, we actually make different proteins, okay? So let's deal within a species. You have one individual human, and you can have another individual human. So between the two individual human organisms, they make similar proteins, but you may also find that due to their genetics, between two individual humans, they can also make different proteins. And that right there is important genetic variation between individuals within a species. And so what your phenotype is, this is basically just review. Your phenotype is a product of the inherited genotypes and environmental influences. You're like, what? So genotype is the genetic makeup. The phenotype is how that genetic makeup is expressed. Why do we throw environmental in there? Because just because you have a specific genotype doesn't mean it always gets expressed the same way. Environment can be very influential on how a genotype is expressed. If you recall from your 111 class, you may have seen the hydrangeas, depending on what pH they're grown at, they can actually express a color of anywhere between a blue to a purple to a pink to a hot pink. And that depends on the pH of the soil in which they are grown. But their genetic type, their genotype, The genetic makeup is exactly the same in all those plants. It's just a matter of where they were grown. So natural selection can only act on the genetic component, meaning it doesn't matter what color they were, natural selection is going to act upon the genetic component. So if the genetic component is not suitable, so for example, if those hydrangeas are not suitable in any of those colors, in any of those components, then they're not going to grow in that area. If it turns out that they need a higher nitrogen content, it doesn't matter if it's pink or blue or purple. So it is that genetics is requiring something else for them to grow. That's a loose example. I'm sure I'll have many others. And so let's look at some phenotypic differences. Some phenotypic differences are determined by a single gene, meaning it's just cut and dry, black or white, what you learned with Mendel, purple or white flowers, okay, in either or basis. But of course, we have learned through life that it's not that simple. Other phenotypic differences can be determined by 2 or more genes, and they can also vary along a continuum within a population. So it could be 2 or more genes, could also be 2 or more genes and the environment in which they are expressed. So human height is a perfect example of using more than one gene. Human height is a result of 3 different gene combinations coming together. Go ahead and stay here for a moment. I'm going to go ahead and find a nice little human height example. So let's go ahead and share my new share. So we have actually here skin color, which is very similar to the human height also. Actually, let's use skin color since it is the same. Skin pigmentation, perfect. Ah, terrible picture. Let's try again. I know we can get a better one. What they call this is polygenic inheritance. This looks pretty good. Or at least right now. You just go straight to this website and do it from there. Okay, so what I'm showing you here is polygenic inheritance, meaning more than one genes, and they come together to make basically a large possibility of how many different shades. And so human height works the same way. I found this example instead. So when you look here, it takes 3 different genes to make skin color. Gene A, gene B, gene C. If you have all of the recessive genes, so recessive A from mom and dad, recessive B from mom and dad, recessive C from mom and dad, then you have the lightest of the light skin. And if you have instead dominant A, dominant B, dominant C, all together, they're going to make the darkest of the dark. So if these 2 individuals come together, what you're going to end up with is an individual that is heterozygous for all 3 of those genes. They have a dominant and a recessive, a dominant recessive, and dominant recessive combination. And you're going to find that their skin tone is that of the intermediate of the lightest of the light and the darkest of the dark. Okay, So if you get this individual together with that is heterozygous for all three genes, with another one that is heterozygous for all three genes, this is the possibility of their offspring. All of these different possibilities. And so one individual, the male will make sperm, and the male can make different combinations of sperm. They can end up with a dominant B, A, dominant B, a dominant C. Look at that, A, B, C. Or dominant A and B, recessive C, boom, boom, boom. I'll let you work out the rest, okay? Same thing with the female. When they make eggs, they can end up with a recessive A, a recessive B, and a recessive C, right down here. Or a recessive A, recessive B, and a dominant C, just like such. You can go ahead and work out the rest. So when the sperm come together with the eggs, you have all of these different possibilities. So for two individuals to come together with intermediate skin and are heterozygous for all three traits, they can end up with offspring that is very, very dark or very, very light. Well, not quite there, very light, okay? Or anywhere in between. Anywhere in between. So I'm looking at this. This chart doesn't look right. This should be a possibility here too. It should be 1 out of 64 possibilities. Oh, I hate that I found you a chart that didn't quite go. So with that, what you will find is a certain see, 1 out of 64. I think they just have a typo. You will find it fits on a bell curve. So you will have the lightest of the light on one end of the bell curve, darkest of the dark on the other end of the bell curve, and in the middle, that intermediate of those. And then you will find that this is going to have the most, and then on each of those sides of the spectrum, the least, just a step down on that, each of those sides, a step down. So you are figuring out the probabilities of what it would be to have each of these different skin tones. The graph for height looks exactly the same. So with that, horse coat color, very similar too when it comes to those phenotypes. Look at this. All of these horse coat colors, almost like they represented the graph that I just showed you. Okay, so genetic variability / nucleotide variability. If you recall, the nucleotides are the molecules that will come together to code for the genes. So your genetic variation can be measured in the gene itself variability, the whole genes, and it can also be broken down to the individual nucleotides, adenine, thymine, cytosine, guanine. Sound familiar? And so your nucleotide variability is measured by comparing the DNA sequences of 2 or more individuals within a population. So we are at a point in time where we can actually look at the genetics and take this down all the way to the DNA sequencing and the nucleotide to look at the actual genetic molecules, adenine, thymine, cytosine, and guanine. So when you think about the phenotypic variation, just think phenotype, the physical, pH. Think about the physical manifestation of the genes. So some phenotypic variation does not result from genetic differences. Like I was explaining to you about the hydrangea flowers, how they can have the exact same genetic makeup, but it is the soil pH which causes the color to be a different color. And so only genetically determined variation can have evolutionary consequence. What this means is it's the genes that's passed down from one generation to the next. It's not the color that is passed down from one generation to the next. The offspring, hydrangea flower, they have to make their own color. And say you take a hydrangea flower that is purple because of the pH soil, and that hydrangea flower has offspring, but instead you plant that offspring in a different pH. That hydrangea flower might possibly be blue. So it is the genes that are passed on, and those genes get to express the phenotype. So check out this non-heritable variation. And so this is your, these are your caterpillars, same type of caterpillar. It's really cool. So this is the caterpillar, and his diet was that on oak flower. Okay, it was on the oak flower. But this caterpillar, he ate upon the oak leaf because of the chemical makeup of their different diets that cause their phenotypic expression to differ. Their genes don't differ. Their genotype did not differ. It is the phenotype that caused them to differ. Did I say that right? I can't rewind it to see, so I'm going to say it again. Just make sure it came out right. The genotype of this caterpillar and this caterpillar did not differ. It's their phenotypic expression that differed because of the chemicals that they were eating, caused them to express different phenotypes. So the sources of genetic variation. New genes and alleles can arise either by mutation or gene duplication. Number two, sexual reproduction can result in genetic variation because it combines existing alleles. So with that, when you get, actually, I kind of want to back this up a little bit. So through mutation, if you recall, let's go ahead and draw my very, very perfect DNA molecule. I say it's perfect because I'm such a wonderful drawer using my pad. All righty, so here is your DNA molecule. I'm not going to sit here and draw all the A's and G's and C's and T's. Might draw a couple for illustration, but that is about it. So here is the DNA molecule. Okay, so when let's go ahead and change this color, go into some black. A section of a DNA molecule, we'll just say this section here, we'll make it small, is going to function as a gene. Meaning your adenine, thymine, cytosine, all of these, depending on the specific nucleotide sequence, I told you I would draw a little bit, thymine, cytosine, thymine, and make it easy. That's the adenine. Oh, that's supposed to be an A. There we go. So ATT, a T goes here, an A here. Does this look vaguely familiar from your chapter 16? Because your adenines pair with the thymines pair with your adenines, the cytosines pair with your guanines, and your guanines pair with your cytosines. Okay, so then with that, this is your gene. Boom. Call it gene 1. So new genes can actually evolve via mutation. If you recall, I drew your, a few slides back, I drew your chromosome, and I showed you a section of chromosome that had an allele on it, okay? The allele is an alternate version of the gene. I have now gotten closer and looked inside that allele, and I see the gene. So that's where we are right here. We are looking into the gene. And so what can happen because of genetic mutation, let's go ahead and erase. Let's say this right here should have been, oh, I didn't mean to erase the whole thing. That's okay. One was a G, one was a C, okay? But through genetic mutation, something happened when those genes were replicated, and something replicated incorrectly. So instead of a G and a C being in that spot, we're going to say an A and a T were instead in those spots. Well, when an A and a T came in that spot, guess what? It's no longer gene 1. Sometimes it might code. If you recall, we went through all of the different codons, and depending on what the codon is, AAT, it would sequence, it would actually, translate into a specific amino acid. Well, we're pretending here that the change caused it to translate into a new amino acid. So hoping it's functional, it's no longer gene 1, but it now becomes a new gene. We're going to call it gene 2. So that is the case where new genes can arise, and that can cause alternate versions of the alleles. So if an organism has a specific color coat, like when you saw the horses had a specific color hair, and you had a genetic mutation, it is possible to cause an alternative version or color of that hair. And so that is a big, broad, loose statement, trying to give you an example of how mutations have happened. Because once upon a time, we were all exactly the same until genetic mutation just kept happening and happening and happening. When I say exactly the same, what I really meant to say is bacteria. Okay, and then it just all snowballed from there. So, alrighty, so formation of new alleles. Probably going to cover exactly what I just said all over again. New alleles, they're going to arise by mutation, like I was just saying, and a change in that nucleotide sequence of the DNA, which I just showed you. So important. Only mutations in cells that produce gametes can be passed on to offspring. Meaning, if I have a mutation on my skin, say I get skin cancer. Why? Because UV radiation has caused my skin cells and the DNA within the skin cells to mutate, possibly causing different nucleotide sequences. So I get skin cancer. That is a mutation. In order for offspring to get the mutation, it has to happen in the gametes. For men, gametes are sperm. For women, gametes are eggs. Okay. So what that means is, if I have skin cancer and I get pregnant during the time I have skin cancer, the child is not going to possibly inherit my skin cancer because that mutation took place in cells that were not my eggs. So that means it is not passed down. So in order for evolution to happen, evolution has to, the mutations have to take place in the cells that produce the eggs. So that is why it is very important when you go to the dentist. The first thing that they throw on your lap when you go to have x-rays is a big lead vest. They throw it on your lap. Why? Because they don't want to possibly mutate, number one, your reproductive organs, and then also on your chest, in your abdomen, you got a lot of really special organs that help keep you alive. So they don't want to be mutating those either. And so this is very important. So I put a star here. You should probably put a star there too. A point mutation is a change in a single nucleotide in a DNA sequence. This example that I just showed you, that is a point mutation. And it actually caused this DNA sequence, this codon, to be a different code. Now, I haven't looked it up. I'm just hoping that I did a nice different code on that one because I threw those in on the top of my head. All righty. So the effects of point mutations, it can vary. It can alter the phenotype to be harmful. That could be bad. It could, the harmful mutations might be hidden from selection in recessive alleles, meaning that just because an individual now has a harmful mutation in their gametes doesn't mean that that mutation is going to get expressed right away. It could be hidden in as a recessive allele. Okay, so mutations that result in the change of phenotype, they are sometimes beneficial, believe it or not. So don't always think that a mutation is a bad thing. Mutations are also what has throughout time helped us to change and evolve. And so a lot of times we always think about mutations as being a detrimental thing, and that is not the case. So effects of point mutations can vary. Point mutations in non-coding regions, meaning introns. Go ahead and review your Bio 111. Introns are those parts of your mRNA that do not leave the nucleus. They are spliced out with the spliceosomes and left inside the nucleus. And so if you have a point mutation in a non-coding region, then that variation is just neutral. It really doesn't cause any change in the phenotype of that organism. So there's no selective advantage and there's no selective disadvantage. So your mutations to genes can be neutral because of the redundancy of the code. If you recall your codon chart, there are 64 different codons. And with that, 3 of those are stop codons and the rest are going to code for amino acids. But there's not 64 different amino acids. Some of those codes are redundant. If you need a review on this, you can refer back to your chapter 16. I'm sorry, your chapter 17. Chapter 17, transcription and translation, and this will be in the translation portion of chapter 17.